really annoying.
Thank you all for coming, and what's really cool is that there's at least 3 people here, so maybe not quite half of you that are in this talk with photos. But this was a really fun exercise for me to put together this talk because it's giving me an opportunity to talk about a few of these projects that have been either finishing up or kind of newly finished up or starting. I've typically been focusing a lot of my talks on the Hatchery Wild Study and some of our other herring work. So the talk today is going to focus on results from our juvenile herring study that came out earlier this year as part of the EVOS herring program. I'm going to talk about kind of a side project related to the Hatchery Wild Study, so not at all about the Hatchery Wild Study, but a side project that we had funded through the National Fish and Wildlife Foundation, and then also a recent project that Marion and I started this summer on tufted puffins with funding from NPRD. And obviously all of these projects are related because we're using stable isotope methods to infer aspects of the feeding ecology of all of these animals. So that's kind of going to be the theme that runs through the talk. And I guess one of these old sayings that we all know, you are what you eat, and this really speaks to the idea of feeding ecology. And one of my sort of longtime favorite comics about animals eating each other is this about leopard seals sitting at the Thanksgiving dinner saying, oh, it was lovely having the Adelies over for Thanksgiving. Because we all know leopard seals eat Adelie penguins, and of course, the Adelie penguins are not there anymore. So thinking about food webs and trophic ecology, this is kind of your classic textbook example when we think about different critters in a food web that are all linked together because they're sort of eating each other, right? On the bottom here we have phytoplankton. This is typically like the base of marine food webs, primary productivity, that are basically consumed by zooplankton, and then upper level critters like forage fish, and then marine predators, whales, birds, and seals. And really, food webs are really a core thematic framework in the ecological sciences. This is something that really translates through all different kinds of research in ecology. And feeding relationships really can provide insight into almost kind of any area of ecological research, from population dynamics to the cycling of nutrients through ecosystems. And so this was just taken from a review paper that gives a little bit of the history about food web ecology and science, and really it kind of began in the early 1900s, way back 1925, with some work by Charles Elton. And then what you can really see is that the field has kind of exploded in more recent years, particularly from the mid-90s onward. And a lot of what I've been interested in is using stable isotope methodology to look at aspects of food web structure, but also kind of ecological relationships and things like that among marine animals. So just to give a definition in terms of what we're talking about, food webs are networks of consumer resource interactions among groups of organisms, populations, or aggregate trophic units. And one of the really sort of inspiring things that I found about this field of research was just starting to think about energy flow through food webs. And this is something that I started to think about actually before I ever went to grad school. And I was actually reading some work out of this paper, or actually this is a book by Cynthia Carey, Avian Energetics and Nutritional Ecology. And the first chapter is by Bob Ricklefs. He's a very famous ornithologist, and he's done a lot of work on the energetics of breeding, especially in seabirds. And what he said was that energy is a universal currency and standard of comparison in avian biology. Every biological structure has an energetic content. Every physiological function and activity requires the metabolic transformation of energy. Energy and power have been subjected to a variety of measurements which partly characterize the physiology, behavior, life history, and ecological relationships of organisms. Okay, so this just got me so excited because thinking about how all these different species and critters are interacting, and that there's this one universal currency that kind of connects all of them, which is energy throughout food webs. So as I said, I got interested in this sort of before I went to grad school. This is Dan Roby. He's a professor down at Oregon State, and he does a lot of work with seabirds and energetics ecology of seabirds. And this is really where I sort of got an introduction into physiological ecology. And then I went on to be a grad student with Dan Essler. Dan is a PI in the Gulf Watch program here with us at the Science Center. And I also worked with Tony Williams at Simon Fraser University. And so this is actually me as a master's student when I had long hair working on ducks out in the Waiheke Delta. I went on to work with Bill Fraser down in Antarctica on penguins. And so these were sort of all of my experiences that shaped me before I got to the Science Center and started thinking about these projects that we wanted to do here. So studying animal diets is not always straightforward. Typically, if you're a seabird biologist, one of the things we do is spend a lot of time out in a blind or something and looking at the types of forage fish that a seabird might bring back to its chick. And this is one way to study diet. Another way to study other seabirds' diet is to look at what the parents are provisioning chicks, and they actually regurgitate it. So for species that aren't just bringing back whole fish, sometimes you actually have to stomach lavage these animals and look at what they're bringing back. So this is a sample from an Adelie penguin looking at all the different little tiny forage fish heads. So we pulled otoliths out of these fish heads, and you can actually tell the species of fish that Adelie penguins are foraging on. Kind of another alternative way to look at diet is to look at scat, okay? So you can look at otoliths in kind of the poop of different animals, and some people are actually using eDNA to look at what's being pooped out by these different animals. So these are kind of your traditional standard ways of studying diet in animals, but the real kind of problem with it is that the diet studies only give a snapshot in time of what these animals are eating, right? So we would only kind of see what the puffin's bringing back at that one time, and we don't know actually what it's consuming itself. So it doesn't really give a very strong picture of what diet is. Stable isotope methods in food web ecology have really allowed us to kind of get around that problem. And I'm just going to call it SI analysis for stable isotope. And it's really emerged as basically one of the primary tools to look at trophic relationships and the flow of energy through food webs, okay? And this was really one of the seminal papers about this in terms of applying these biogeochemical signatures to ecology. This is Peterson and Fry, and they published this paper in 1987 in the Annual Review of Ecology and Systematics. And kind of leading up to this, there were some other seminal papers by De Niro and Epstein in kind of the late 70s and early 80s that talk about the influence of how diet changes carbon isotopes in animals or nitrogen isotopes in animals. And so stable isotope analysis is really based on, primarily the literature is going to look at just a handful of elements. So this is carbon, nitrogen, oxygen, sulfur, and hydrogen. And they all tell us something different about and what an animal is consuming and how it's sort of moving through the landscape. And most of the work I've done has looked at carbon and nitrogen stable isotope variability. So that's what all the examples are going to be about. But just to understand, there are different signatures that you can look at, and they kind of tell you different things. So one of the great things about stable isotope methods is that they provide a time and space integrated insight into trophic relationships among organisms. And so hopefully I'm going to try to describe what I mean by time and space integrated insight here. You can imagine that we have these elements of carbon and nitrogen that are like flowing through the food web from primary producers all the way up to top predators as they kind of eat the lower trophic levels. And for elements like carbon and nitrogen, and especially nitrogen, for example, as this element moves through the food chain, the ratio of these heavy to light isotopes changes. And so animals actually excrete the heavier isotope. So as it moves through a food web, what you do is you get a different ratio of the heavy to light isotope. And we can measure this in the tissues of birds. For carbon, it actually tells us something about primary productivity. So different plants have different photosynthesis pathways. which results in these different fractionation values for carbon. And so that's kind of the gist of it, is just that these ratios of the heavy to light isotope change throughout food webs, and we can use that as a way to kind of mark and track aspects of how energy flows through a food web. So in terms of the time integrated insight, you can imagine like where we did a project on pink salmon with stable isotopes. We go out and collect pink salmon in the spawning streams, and we're going to measure carbon and nitrogen isotopes in different tissue types of the pink salmon. So for example, we measured it in liver and in muscle and in the gonad. And so muscle tissue actually has a longer turnover rate than liver, because liver has a lot of blood in it, right? And blood is a type of tissue that is very metabolically active, so it turns over this isotope signature very quickly. And so because of the sort of metabolic activity of the tissue, it can give us different windows of understanding of when the diet was changing in these animals. So by sampling different tissues, it can give us an idea of different time frames that the animal was foraging. And so what we do is we sample these tissues, we dry them in the lab, grind them up into literally like fractions of a milligram amount, and then combust them in this machine. And what it gives you is this delta value right here. And it's just the ratio of the heavy to light isotope in the sample compared to that what's in the standard, -1 times 1,000, and you get your data. Another way to think about this is in terms of this space integrated insight into how animals are foraging, and actually how they're sort of connected throughout different stages of their life cycle. So this is from a review paper by Rubenstein and Hobson. This is Keith Hobson. He is kind of one of my heroes, and he really advanced all of the stable isotope approaches, particularly in avian systems. And so you can imagine you've got a bird that's got a different kind of breeding area from its wintering area. And if in the wintering area, you know, it has a couple of different habitats and it molts its feathers, it's going to grow its feathers from either habitat A or habitat B. Then when it goes to the breeding ground, it's going to carry these isotope signatures in its feathers all the way to the breeding ground. So you could sample birds on the breeding ground and know something very specific about where they were wintering, and look at these kind of migratory connections between wintering habitat and breeding habitat, which is really powerful because it allows us to kind of understand potential drivers of population change, whether it's happening, you know, in the winter or in the summer, and piece together things that way. This is Samantha Franks, and she was a friend of mine in graduate school, and she did work on sandpipers. This is from Western sandpipers. And basically what she did was she looked at migratory connectivity across kind of breeding areas up here in Alaska, and then a number of wintering areas. So what they did was they went and sampled sandpipers down in the wintering areas for their feathers, established kind of the variation across these different wintering areas. Then they went up and sampled sandpipers in Alaska, and they were able to basically partition the proportion of the birds that were sampled across these different wintering areas, across these different regions from Russia and sort of the western part of Alaska. So this is like amazing, right? Like these birds are traveling like thousands and thousands of miles, and just by sampling them on the breeding area, we can know something about what wintering areas they're using. So really, I can't kind of overstate the fact that isotope methods have really revolutionized how we can do animal ecology, which was my next statement there. And so one of the things I've been excited about is kind of relating the stable isotope variability to factors like reproductive performance and survival, and linking them with environmental features. And so this kind of brings me to the 3 different study systems that I'm going to talk about for the rest of the talk. So herring, pink salmon, and tufted puffins. And so what I want to try to do is just explain how we're using isotopes in these different systems and what we can learn about it. For the herring work, this is a paper that just came out And we all celebrated Scott's birthday with mustaches and Hawaiian shirts yesterday. And then Mrs. Megan Roberts, who was an amazing assistant on this project. She worked both with Tom and myself. And so what we were really interested in is looking at variation in winter conditions, so kind of like the energy content of herring, in relation to sort of oceanographic exchange with the Gulf of Alaska. And we were using stable isotope methods to do this. So many of you know the herring story here in Prince William Sound. This is from a paper by Melissa Muradian, which came out in 2017, and it's her model showing the biomass estimates over time for Pacific herring. And so what we know is that the population crashed after the Exxon Valdez oil spill, and it's been at these very low levels for a very long time. And so the Science Center has long-term funding through EVOS to really study why this population hasn't recovered from the spill. And one of the things that we're interested in is factors about the young fish and what helps them to recruit to the spawning population or not. So we sort of took an energetic approach to this question, and we actually had data back in starting from 2007 when Tom first started this project. And really, we were interested in looking at the overwinter energetics of these fish. And so this is data from 2007 through 2015, essentially in November. That's the gray bar. And March is the blue bar. And what you see here is that always, you know, the November bars are larger than the blue bars, and that's just showing you the whole body energy density of these fish. And so the idea here is that juvenile Pacific herring have to pack on energy in the fall to get through the winter to survive, because they don't have a lot of food in the wintertime to eat. So when they you know, emerge, not really emerge, but when they're around in the spring, they're in a much reduced body condition. And we know that this actually stays low throughout the whole year. So in November, energy levels are high, and then they kind of fall, and they basically just fall throughout the winter through March. So what we did with this paper was we actually kind of used some of Tom's earlier work. This came out in 2009, where he was looking at kind of stable isotope gradients in the northern Gulf of Alaska versus Prince William Sound in a various number of zooplankton. And so they went out and they sampled across all these regions in the Gulf of Alaska, and then they had some interior sampling areas inside Prince William Sound. And what they found was that there are isotopic gradients that happen between the Gulf of Alaska and Prince William Sound. So on the y-axis is our delta carbon signature, so carbon stable isotope signature, and then nitrogen. And then these stations, basically like A, B, and C, are out here in the Gulf. H, I, and J are somewhere in here, I think, inside Prince William Sound. Well, G, M, O, G, R. And so what you see is that carbon is basically more enriched out in the Gulf of Alaska than it is in Prince William Sound.
Backwards.
Sorry, the other way around. Less enriched. Yeah. Less enriched less enriched in the Gulf of Alaska, more enriched in Prince William Sound.
I use depleted because it's more negative in the Gulf, basically.
Okay. But anyways, the point is that there are these isotopic gradients, and we can use this kind of to our advantage. The signal was not as evident for nitrogen, so we really were mostly interested in carbon. We looked at both isotopes. So back to our paper, we basically tested this idea that oceanographic exchange between the Gulf of Alaska and Prince William Sound controls juvenile herring nutrition and the quality of the young fish. And this might actually play a role in how well these fish recruit to the spawning population. So we went out for many years and sampled in November and March from different areas all throughout Prince William Sound. And so the November sampling is in the circles, and the March sampling is in the triangles. And what you see is we have sort of four kind of ecoregions here in the Sound. This is like the eastern region in yellow, the northern region in white, the western in orange, and kind of this central area in green. And what's shown on this map is actually the bathymetry of the Sound. And what we see is that there's this large marine trough that comes in and kind of works its way over to the west. So there's different regions of the sound that can reflect oceanographic signatures from the Gulf of Alaska, because this is how water moves into the sound and circulates around. So what we wanted to do was put together some models where we were trying to predict the whole body energy density of these young fish, which we determined actually with the stable isotope methods. But we got energy density from that. And we're looking at it in relation to things like fork length, the carbon nitrogen signature, the region of Prince William Sound, the year of the study, and like an interaction between the isotope and the region. And we did some modeling where we were controlling for the collection bay in this analysis. And so these actually were all the top models that were supported in the work. And so you can see in all of these there was a role for isotope variability in predicting whole body energy density, but also region came out in a lot of these as well. So what this sort of looks like is this. So this is, these graphs are just kind of graphical models of those, of the best supported, graphs of the best supported model. And we've got the 4 different ecoregions of Prince William Sound across all of our different years. And what you see is that this relationship between carbon and whole body energy density is a lot stronger in the north and in the west than in these other regions, okay? So that's interesting. And we also found that in terms of the nitrogen variability, we did see a strong relationship in the northern part of Prince William Sound, but not in some of these other areas. We did kind of the same analysis for the fish we caught in March, and you can see it looks kind of scary. There really isn't much going on. And basically, we think this has to do with the fact that fish aren't feeding that much throughout the winter, and we kind of see that in the data. And I also would just note this kind of crazy wonky line, this was 2016, the spring of 2016, which followed the blob out in the Gulf of Alaska, which I'm going to talk a little bit more about. So, you know, I think we can detect kind of these weird anomalous things using these methods, and I'll talk a little bit more about that later. So I guess, you know, sort of the take-home message is that because we know that there are these isotopic gradients between the Gulf and Prince William Sound, we see that areas in the Sound that reflect a Gulf of Alaska signature resulted in fish being more energy dense as a young fish. So what this just tells us is this is a really cool environmental driver that's sort of structuring the quality of these young fish, and it could have a role in whether they recruit. That's not part of this analysis, but this is just kind of using isotopes to ask these questions about connections between these different bodies of water and what it means for the quality of the fish. I'm going to transition to the next part. Does anyone have questions about that? Or maybe we should wait. Yeah.
I just have a question about the signature on the areas that you felt showed the Gulf.
Back.
From that, the chart.
Oh, here.
Yeah. Is that from them, or do you have any idea, from them feeding outside and then they come in, or is the water coming in?
We've always thought about it in terms of the water being a transport for critters that are from the Gulf that then get into Prince William Sound. And those critters happen to make the fish a better quality. And they're carrying that Gulf carbon signature with them. Yep. Anyone else? No? Okay. So I'm going to go on and talk about our pink salmon work. So this is a study, I actually presented this talk at the American Fisheries Society regional meeting last spring in Anchorage. And this was kind of a pilot study or a side study that we did in association with the Hatchery Wild project. And we got money through the National Fish and Wildlife Foundation, and so this is Pete Rand and Eric Knudsen, who I work with on the Hatchery Wild project, and this is Julia McMahon, who has been a very important help as a technician on this project, and she's now a grad student at the University of Alaska in Fairbanks, and she's going to use this data set in her thesis. So this will be part of her thesis work as a master's student. So I know a lot of you know about the Hatchery Wild study, but just to kind of recap that, we've been interested as part of that project in quantifying the fractions of hatchery pink and chum salmon, both at the Prince William Sound ocean entrances and on spawning streams. And there's another component that Fish and Game is working on, looking at survival of offspring using genetic pedigree analysis. So really the first part is just quantifying spatially the fraction of these fish across Prince William Sound and in the ocean entrances and on the spawning streams. But it doesn't really tell us much about what might be some of the mechanisms that are driving possible differences in reproductive survival, which is kind of the point of the second part of this pedigree study. They want to know if offspring from hatchery or wild parents survive better, but it doesn't tell us like what the mechanism would be. Why would that be? So this study kind of used the Hatchery Wild field campaign as a leverage for trying to look at some of these questions. And basically, you know, we were just really interested in if there are differences in timing of migration, reproduction, body size, or foraging that might set the stage for variation in performance. So things like body condition, gonad weight, fecundity, and egg size that would kind of ultimately shape the fitness of both of these ecotypes of pink salmon. So using stable isotope methodology, it has a long history in salmon, and this is just kind of some of the data. These are the carbon and nitrogen isotopes in different species of pink salmon, and these are 4 different studies. And what it kind of shows is essentially different species are kind of more overlapping in their ocean foraging than other species. This is king salmon here, this is coho, and then these are the pinks, chum, and sockeye. And so, you know, there's definitely variability across the studies. Some of the species are more spread out depending on what study you look at, but then, you know, in some of these other ones, you see there's a lot of overlap between different species of salmon. Other people have looked at isotopic values in the scales of sockeye. In this study, Scatterfield and Finney, they looked at it in Kodiak sockeye. And so they were able to get data all the way back to the 1960s and looked at isotope variability across years and how it changes in response to kind of these big climate patterns and things like that. Out in Bristol Bay, so this is the UW group, they looked at actually how carbon and nitrogen isotope variability is related to body size. I think these are in sockeye, yeah. And what they showed was that there's really strong relationships between the carbon and nitrogen isotope variability and the size of the fish, okay? And so that was true across all ages. So these are age 2 males, age 3 males, age 2 females, age 3 females. And the dots are different populations of sockeye in Bristol Bay. And so what you see is the populations that are really large have different isotope signatures than the populations that are really small. And these were sort of the types of relationships I was interested in with our study on pink salmon, and especially things like this, where we have carbon and nitrogen isotope variability relative to whether an individual fish is from hatchery origin or wild origin. And so this is some of Tom Quinn's work on steelhead in the Columbia River. And basically what he's showing here, the hatchery fish are in black and the wild fish are in gray. And these different ecotypes are kind of segregating in terms of their foraging. Similarly, they saw kind of the same thing, that different size fish, so the length of the fish, is also related to the carbon and nitrogen isotope variability in the tissues, and that relationship changes whether the fish are from hatchery or wild origin. And so these are the things we wanted to look at with the pink salmon data. So we sort of took a first crack at it using basically a null hypothesis. We just said hatchery origin pink salmon are similar in their at-sea foraging strategies to their wild conspecifics based on the stable isotope values, and therefore they're going to be similar in their migratory timing, body condition, and spawning performance. It was kind of a basic approach, but What we did was, and we did this work in 2015 only. We weren't able to do it across a number of years. But we collected pink salmon as part of the ocean test fishery, which collected fish at these different ocean stations in Hinchinbrook Entrance and in Montague Strait between June, July, and August. And we sort of had early and late cohorts for each month. And we collected about 40 pinks per sampling event, so 20 males and 20 females. And then on the spawning streams, we did kind of the similar exercise, but we collected them in August and September. We had early and late cohorts for each stream, and again, 40 pinks total per sampling event, so equal numbers of males and females. So we did a lot of the lab processing for the isotope work in 2016 and 2017. And I kind of already showed these figures, but we basically collected fish, we dissected them, collected tissues from the liver and the muscle and the gonad. We collected otoliths to learn about its hatchery wild status, and this was part of the Hatchery Wild project. So, we used those same data. We didn't take extra fish. We processed them in the lab at the Science Center, and then sent the data to the University of California at Davis that has an isotope facility there. So just to kind of give you an idea of what some of the data look like, this first graph is looking at the proportion of hatchery fish in both the ocean and stream data sets. Okay, so this is using the otolith data. What you see is for our different sampling events from like June, July, and August, the proportion of the hatchery fish increases later in the season. So it just means that hatchery fish are migrating through the ocean entrances later than the wild fish. And then when we looked at these proportions on the stream sampling, what we saw was actually our data set, which was relatively small, only captured just a fraction of hatchery fish across these 11 streams that we worked at. And stream 8, that's actually Comstock. It has a huge number of hatchery fish, so we were happy to sample there because it gave us some contrast in our data set. But specifically in terms of some of the isotope results, in this analysis, we were basically looking at body size of the pink salmon. Okay, so we just developed a PC score for body size. This is really, I think, the length and height of the fish. And we just develop into one score with kind of positive numbers being a larger fish, negative numbers being a smaller fish. And what you see is that in the ocean sampling, the fish are largest coming through early and they're smallest coming through late. And this was in 2015 only. And we know this is maybe not super surprising because we saw that there were more hatchery fish returning later in the season. And so, not surprisingly, hatchery fish ended up being smaller in our data set than wild fish. So this was actually a significant factor predicting body size variation. When we looked at kind of how the size of the fish was related to variability in the liver carbon signature and the muscle carbon signature, we saw pretty significant results. So what this tells us is that larger fish are foraging differently than smaller fish. And that's true for these two different tissue types, and liver gives us a more immediate signature of foraging versus muscle. That's probably capturing several months out from when they arrived in Prince William Sound. When we looked at this for nitrogen, we kind of found some of the same results. There was a significant relationship between the size of the fish and muscle nitrogen signature, but not with liver. When we went on to look at aspects of body condition, okay, so this is like how fat a fish is. This is not how big the fish is. We were looking to predict weight of the fish, but we included size as an explanatory variable, so we're controlling for size. And you would expect that larger fish weigh more, okay? So we remove this relationship from the model, and then we look at what the effect is for these other variables of interest. And so what we found, actually, so in terms of condition, that the best conditioned pink salmon in the ocean test fishery were coming through in the middle of the season. So this was kind of in July, with the June and August fish being of less condition. We didn't find any difference condition between hatchery and wild fish. Even though the wild fish were smaller, there was no difference in their actual body condition. And actually, interestingly, we found really non-significant results between the isotope signatures of these different tissues and the condition of the fish. So even though we think size matters for foraging, it doesn't seem to matter with the condition of the fish, if that makes sense. I'm going to talk a little bit about the stream sampling.
Before you go,
yes.
was there any evidence that they had fed less towards the end and that was why their condition was decreasing? There's less food available later in the summer, so they may be already starting the starvation while they're in saltwater before they come inshore.
It's possible. We didn't really do any stomach content.
But I mean, from the liver analysis, anything that stood out, like C to N ratio, anything that,
No, I mean, honestly, we didn't look at that. We should, and that's something that Julie can do, but we haven't looked at that yet. So, thinking about the stream data set. So in this analysis, we're looking at body size of the fish on the streams. And kind of similarly, we found that the early fish arriving to streams were larger, structurally larger than the later fish. And we also found that wild fish were larger than hatchery fish on the streams. So this actually follows really closely what we found in the ocean test fishery results. In terms of looking at the size of the fish on the streams, we kind of found some similar relationships. So for example, I think the relationships were stronger for mussel carbon and nitrogen than for liver, which I want to talk to you about actually, because we had some different results in the ocean test fishery. But anyways, so then we went and looked at condition again in the stream fish. Again, we're just controlling for this size, we're controlling for the relationship between size and weight of these fishes. And then afterwards, we can say that the early fish in the streams were of higher condition than the fish that were returning later, but there was no difference in condition between wild and hatchery fish. And actually, we found basically no relationships except for that one, and that's kind of a weird one. Well, sorry, there was something there, but these relationships were not as strong in the stream fish between the liver and mussel isotope signature and condition, which was similar to the ocean results. So kind of just sort of the take home of all of this is that there were really important results between pink salmon body size, run timing, and hatchery or wild origin that correlate with stable isotope variability, but that wasn't true for condition. And really this is just kind of a first glimpse into kind of understanding trophic ecology and how it might affect aspects of the evolutionary ecology of these fish. And we think that this is really actually an important study because foraging competition at sea is a huge aspect of ocean ranching in Alaska, and it's something that we're not studying, and it's something that I would like to try to turn into more of a multi-year study, kind of stemming from some of this work. The other thing I'd like to do, I said this is going to be part of Julia's thesis, is look at these kind of same relationships, but across different species. So probably between pinks, sockeye, and maybe chum for the same year over multiple years. So that's kind of a recap of the pink salmon work. Does anyone have questions about that?
I'm just trying to think,
Yeah.
why are hatchery fish smaller than if they're just as good a condition? I'm just trying to sort of.
natural selection or lack of it in hatcheries.
So you think it's just that?
It's the behavior in the stream because the alpha males are bigger and they contribute more of their DNA to the offspring. And so they're passing on the largeness gene, whereas in hatchery they do a total random spawning and they're getting rid of it. And this has been seen in salmon in the lower 48 as well. They're becoming domesticated.
So the hatchery fish are always
They're
smaller.
stunted, their teeth are getting smaller, basically. That's really probably the more negative thing about the hatcheries is we're getting rid of the strengths that take place because of natural selection that's going on in the streams.
Okay.
Just like with birds and everything
So
else.
have they
they been measuring the fry then? To see, I mean, so, you know, if the hatchery fish are smaller as adults, then if it's, you know, then one would think that they'd be smaller as fry.
I mean, yeah, that's an idea that you could test. I am not aware of any work, I mean, there may have been, but, I mean, I think there's probably maybe some aspect of a lot of hatchery fish getting released around the same time, and so you can imagine that they're all going out to feed and there's just like tons of them, so that initial growth of their early life is among a lot more competition than maybe natural.
The problem with that question is smaller females often have slightly smaller eggs. Smaller eggs could yield smaller fry, so you might actually be looking at effect from the smaller female size. But I mean, that is a very difficult question to answer because you have to collect, you know, a large number of eggs and fry. It could be done in a hatchery, but and then you need, and the difference might be very small, so you need to have, you know, a pretty large sample to be able to see the difference.
Okay. Any other questions? So, I'm gonna talk just a little bit about, our tufted puffin project. And this is something that, we just started this past summer. And we're working, obviously on tufted puffins. Here's the mighty tufted puffin. And so Marianne and I, put this project together, and we're working with Ann Harding on the outreach component. These are some of the awesome masks that she just made, to work with students on the Puffin project. And then Ann Schaefer is an assistant at the Science Center. And so we're working out at Middleton Island. Middleton, if, I don't know, most of you probably know, Middleton sits out in the middle of the Gulf of Alaska. And it's a pretty interesting place because it's right at the edge of the continental shelf, and there's lots of seabirds that nest there, and it happens to have a very large population.
Are there still a bunch of rabbits running around there?
Yeah. It has a large population of tufted puffins nesting there. And so what we wanted to do was try to understand the annual pelagic distribution of puffins in the Gulf of Alaska. And part of why we were interested in doing this, there was a paper that came out in 2017 by Holly Goyert and co-authors. And basically they looked at kind of changes in the carrying capacity of a number of Alaskan seabirds. And one of kind of the red flags that came out from this analysis was that tufted puffin populations in the Gulf of Alaska, and this is across 3 different population models that they ran, all show that they're going to be declining in the future. And so this is not something that has been true for tufted puffins in Alaska. It has been true for populations in Washington and California, but we always kind of imagined that the Alaska populations were really robust and stable, but this analysis is kind of telling us to think otherwise, particularly into the future for puffins. And one of the things about puffins is that most of the information we know about them is from their breeding season. So seabirds are really easy to study during the breeding season when they come to land to, you know, raise chicks. And that's primarily, like, all of our knowledge around puffins is during the breeding season.
Is the population, or are the population models based on breeding? The number of eggs and things like that? How do they actually?
Yeah, I mean, I think that's when we have the best survey data for
So actually
puffins.
it's a kind of a breeding graph there more than actual population.
Yeah, this is actually, you're right, this is about breeding individuals, yeah. And there actually has been no work trying to track tufted puffins in the wintertime, so we don't know where they go. There's some sort of bycatch data that suggests they're going out into kind of the central Pacific, but we really don't know much about where they go in the wintertime. So what we're using is actually these Geo Locator tags, and Geo Locators, these weigh, what do they weigh, Marianne, like 2 grams?
2 grams or less.
Yeah, they're very small, and we can put them on plastic bands that we put on a puffin. And so the puffins carry these tags around from breeding season. We put them out in the breeding season, they'll carry them around and in theory come back to the same burrow that we find them at. So this is me this summer grubbing in a tufted puffin burrow looking for the adults. And when we found an adult, we would tag it with these geolocator tags. And the geolocator tags are archival, so you have to get the tag back to get the data. And they use light levels, so variation in light levels across essentially longitude and latitude, and you can use these algorithms to figure out where the birds go. So we went out and worked at Gold Hill out here on Middleton Island, and we were searching for puffins, and we marked their burrows, and we'll go back and try to grub in these same burrows to try to find the adult next year. And one of the things that we're doing as part of this work, so we'll in theory know something about where they go in the wintertime, but we're also going to measure stable isotopes in the feathers of the puffins and also blood tissue that we took, which give us sort of two different time frames of understanding diet for puffins. So that's kind of just like a snapshot of tufted puffins. I don't know, Marianne, did you have any other things?
What kind of fish do they eat mostly?
Well, so this is the interesting thing about puffins. They feed their young, like little forage fish like sand lance and
Herring, how important is herring?
I don't think it's that important actually.
And if they find salmon, will they feed them salmon?
I think if they'll, ultimately if they can find anything, they'll.
If all the hatchery salmon, I wonder if they're feeding the chicks the hatchery salmon.
Well, I don't know about the timing. Like they're raising chicks in July.
Well, that's kind of late.
Yeah.
But the fry moving out, in fact, I drove a boat out to Middleton Island, there's salmon popping on the surface, you can see them
really?
driving at night, so
Huh.
they could catch salmon on their way out from all over the place, crossing the shelf. But I mean, you would know that, right, when you see what's in there, what they're eating?
Yeah, so Scott Hatch, who does all the seabird work out at Middleton, he's been looking at diet of a number of different species, including puffins, so he, I'm sure, has pretty long-term information on that. But they feed forage fish to their chicks during breeding. But there's some suggestion that actually puffins are foraging more on zooplankton outside the breeding season. So, forage fish populations kind of tend to have lags relative to kind of climate variables. But so that's why we were kind of interested in this, because if they are feeding at sort of lower trophic levels, it might mean that they're a little bit more vulnerable to ocean changes outside the breeding season. So we're going to compare the feather isotope signatures to the blood tissue, and that'll let us look at the differences kind of between early spring and early summer diet in the puffins.
How deep can they dive?
I actually think they dive kind of deep, like deeper than you'd think.
So they get you thousands without any
Yeah, I
issue.
think so. And one sort of interesting thing, we found an abandoned egg out on Middleton this summer from a puffin burrow, and it was cracked, and you could see that the yolk was a bright orange yolk. And so birds that feed at kind of lower trophic levels, like macaws and these sort of orangey krill type, critters, have the same carotenoids in their tissues. So seabirds that have very orange yolk are probably foraging on lower trophic levels. So it'll be interesting to see what we find. So I guess I just wanted to wrap up a little bit with kind of why we should care about food web interactions in the Gulf of Alaska. We've looked at a number of different critters, both in Prince William Sound, out in the Gulf of Alaska. And really, the issue is that what they find in the ocean is what's sustaining them. And we know that that this is the blob, this is the marine heat wave. And I found this paper in Nature Communications just actually over the weekend. It came out in 2018, earlier this year. And it was amazing. What they said was that the North Pacific blob, the North Pacific marine heat wave that occurred in 2015 and 2016 was the largest documented marine heat wave ever on the planet, and it happened here. And we know that it was associated with extreme die-offs of common murres. In 2015, of course, that was a huge return year for pink salmon, but it also kind of followed the blob years, and a lot of the fish were small that came back in that year. So these are having really important implications for a lot of the critters that we fish on and that we eat and that we bird watch. And so I think that these issues of looking at food web interactions is really an important research area that we should think more about. And it's something that Pete and I are thinking about with our sockeye salmon work. Sockeye have been returning to the Copper River in smaller sizes. that we think is being driven by ocean conditions and what it means for their migration and spawning performance in the Copper River. So it's a topic that kind of spans all of these critters, and it's severe. So I will end there, and yeah, that's it.
So they're not attributing the stuff along the equator to the El Nino? Because there was a big El
I think
Nino
a lot of it
couple
had to
years
do
ago.
with
Yeah.
not enough mixing and low wind.
But the El Nino also affected Hawaii.
Yeah, there was an El Nino in there, but I don't actually know.
Caused
how
some
the
coral
timing of
bleaching,
that
'cause I got some
relates.
photographs of bleached coral.
But I was like
Yeah.
blown away reading this paper in a nature journal saying this was like the largest marine heat wave ever recorded on the planet, and it happened in our backyard, and that's a big deal. So.
Probably responsible for this year's bad salmon return in the Copper River.
Yeah. Yeah.
So,
Oh, sorry.
I'm sorry.
Helen.
Oh, there, okay, go ahead.
I think we found out several this past week exactly what sockeye salmon eat and where they migrate. Is that
Could you repeat the question?
what's I was trying to find out for several days what sockeye salmon eat.
You have to ask what stage, because they're
And
planktivores.
in when they go out in the ocean.
They eat everything from, plankton to juvenile nyctophids, lanternfish and juvenile squid. And a Canadian guy did a lot of stomach work on it way back when, I can't think of, oh yeah, Robin Lebrasser. Try to look up his name, you might find some reports done in Canada. He did a lot of, cruises out of, because I remember he gave a talk, they actually got bigger lanternfish in the sockeye than they did in their catches. It was in excuse me. But these were probably sockeye on their way home, so they're, getting bigger and bigger. But they, you start out, they have long gill rakers, they feed on plankton in the lakes, so they start out as strict planktivores and then they move up. And they live in lakes generally for the first couple of years. Although we have some zero checks in system here in town, so they go out right away, like pink salmon. The jargon is called zero checks because they don't have a winter check from the first year, so they're called zero checks. That's just fisheries jargon.
I can maybe send you some papers. Sorry, David,
So, way back in the first part of the first talk, or segment,
Yeah.
the one graph showing the commercial take a herring and then the biomass numbers and the like. But the one dotted line in there with the salmon fry, yeah, there we go, pink salmon fry, is that the numbers of release from hatcheries?
Yeah, I think there is a hatchery component to
That's
that.
the hatchery releases,
Yep.
yeah. Sure complicates it where it's overlaps right on the oil spill there.
Right.
Of their big
Yeah, I don't know,
increase.
did that factor in Melissa's model, or?
Yeah,